All right, so hi everyone, and thanks for tuning in to this week's edition of the Plant Biology Seminar Series. It's my pleasure today to introduce our speaker, Associate Professor Matt Lewsey from La Trobe University, although he's currently joining us bright and early from the UK. I think it's about 7 a.m. there, so thanks Matt for doing that. Matt completed his PhD in plant virology with Professor Carr at Cambridge in 2006, and then continued as a postdoc there studying plant virus vector interactions. In 2010, he was awarded a Marie Curie International Outgoing Fellowship to study plant epigenomics and hormone biology in a joint venture with the ECHA Group at the Salk Institute and Professor Solano at the Spanish National Centre for Biotechnology. In 2016, Matt established his group at La Trobe, where they work to develop and apply omics technologies to study genome regulation in response to different environmental cues across a range of plants and mammals. He has since also become the director of the La Trobe Genomics Platform, co-deputy director of the ARC Research Hub for Medicinal Agriculture, and also served as director of research for the La Trobe School of Life Sciences from 2020 to 2022. With all that said, many thanks for joining us so early, Matt, and looking forward to hearing about what you and your research group have been up to. Take it away.
Thank you very much, Deep, and thanks for the invite. Thanks everyone for having me to speak today. So I will tell you particularly today about our work on understanding genome regulation during seed germination. So I probably don't have to work too hard to convince a room full of plant scientists that the first hours of a plant's life are very important. So when a seed develops, it dries and it packs down and it's essentially metabolically inactive, dormant. It's not doing very much and it's going to hang around until it perceives the correct conditions to germinate. But upon perception of the correct conditions, an awful lot happens in a very short period of time. So you're seeing here an example of an Arabidopsis seed going through germination, and Arabidopsis is the only species I'm going to speak about today, really. So the dry seed perceives the correct conditions, it's going to imbibe water, and this causes the cells to begin to expand. It allows biochemical activity to commence. You'll get the start of respiration, transcription, translation. The expansion is going to continue until the endosperm ruptures through the tester or the seed coat, and then it will continue further until the radicle, the nascent root, emerges out through the seed coat as well. And that is the end of germination in the strict sense, the emergence of the radicle. I'll point out that throughout this, the only growth that's happening is through cell expansion. There's no cell division and there's no cell death, just cell expansion. And so from an applied perspective, this process is super interesting. The seed only gets one go at this, really. And so if it does this incorrectly, the plant's life is over. It's not gonna do anything. And so growers and producers need this to happen correctly at the right time in the correct conditions for them to be profitable. But from a fundamental perspective, it's also quite fascinating to think about how all these different processes are controlled, how you go from something that is inactive, metabolically inactive and doing nothing to extremely active in a very short period of time. One of the things, or the thing really that I personally find most interesting and that this talk is going to focus on, is the fact that during this process, the genome has to reactivate. And the reason it has to reactivate is that during seed development, the nuclei of the cells in the embryo pack down. So they literally get smaller and DNA is packed into heterochromatin. And it's thought that the reason for this is to protect the DNA when the seed dries out. So it helps it maintain stability and not get too damaged. These images illustrate this process. So this is work from Wim Sop's lab, and in what you're looking at are pictures of a nucleus from an embryonic cotyledon cell. The nucleus as a whole is stained using DAPI, so it's blue, and then we've got DNA methylation stained in green as a marker in there. On the left-hand side of the screen, you can see seed development. You can see the nucleus is contracting between 10 and 20 days after pollination. It's getting smaller. And at the same time, you can see that the DNA is moving, so that you can see the green signal moving. The DNA is moving into chromocenters. It's taking on a heterochromatic state. Then as you move into germination, you have to reverse all of this to permit gene expression so that gene expression itself can start. It's necessary for gene expression to start to specify all the functions encoded by all the different cells in an embryo as it grows into a seedling and onto a plant. So you can see on the right-hand side of the screen, the nucleus is very small at first at 2 hours. It becomes larger, the DNA spreads out at 24 hours and continues through to 72 hours. So today I'm going to talk to you particularly about the process of gene expression, what happens to gene expression as the genome wakes up. So I'm not going to talk about how the nucleus goes about expanding and becoming, losing its heterochromatic status. I'm going to talk to you about the events downstream as the genes are turning on. What are those genes doing and how are they controlled? So the outline of my talk looks a bit like this. First, I will speak to you about transcript dynamics and temporal regulation of the genome during germination. So those events as germination progresses. And all of this is going to be in whole seeds, essentially. This is all work that we published a little while ago and is out there that you guys could read, but I didn't want to just present work we'd already published to you. So at the end of that first section of the talk, I'll explain a limitation in our prior study, and then I'll talk about how we're tackling this by using single-cell transcriptomic methods that allow us to understand the cell specificity of genome regulation. And that's all entirely new work. It's still a work in progress, but I hope that you will find it thought-provoking. Let's get started with the transcript dynamics and temporal regulation. So when we started this study, what we really wanted to know was how does gene expression change over time? We wanted to get a really tight understanding of this because you need to see how it's changing in order to understand how that's being controlled. We set up an experiment like this. So as I said at the start, in Arabidopsis, our experiment took seeds at different stages of germination. So all the way from the dry seed at harvest through 0 hours prior to the start of the experiment. Then the stratification phase, the cold and the dark, which is quite common when working with Arabidopsis, and transitioning them into the light and at room temperature, which really allows germination to move forward. And so we studied 9 time points, I think that adds up to on here, across germination. At each time point, we've taken samples and we've analyzed gene expression. We've analyzed whole transcriptomes. We also analyzed genome-wide DNA methylation, and we analyzed small RNA abundance as well, but I'm going to focus on the expression data today. So the first thing that we did was ask, all right, which genes change across germination if we've measured gene expression at all of these time points? And the short answer is nearly all of them. Almost the entire genome is differentially expressed during germination. I'm showing you a representation of that data on here. So on this slide, time moves down. This is a heat map with red indicating high expression, blue indicating low expression. Our dry seeds are at the top row, moving down through stratification into the light, and the final 48-hour time point in the light in the bottom row. There are about 27,500 genes encoded by Arabidopsis. And over the course of germination, we find that more than 24,000 of those are differentially expressed in some manner. The differential expression broadly falls into 3 categories. The first is a transient upregulated category labeled in the center of the heat map there. These genes expression of these genes goes up around about the transition from the dark into the light, and at a broad level, they encode functions that might be associated with the things you need early in germination. So energy, metabolism, starting up, things like that. There's a second group of transcripts that are late upregulated on the right-hand side of the heat map there. These guys encode a lot of things associated with photosynthetic activity, for example, things that you would need towards the end of germination when the seed's transitioning toward becoming a seedling. And then on the left-hand side of the screen, our third group, this was perhaps the most interesting and perhaps the least expected group. What these are transcripts very high right at the start of the experiment, so in the dry seed. And these guys start high and they go down. They're mostly being degraded away. What these represent are transcripts that were actually produced. They were transcribed during seed development and stored stably in the seed throughout its dry period. They are actually, later work has shown, they are packed into polysomes. They are into granules in the cytoplasm and stored stably there. And they presumably encode functions, proteins that are needed very early in germination, that are needed before transcription itself has gotten going in the developing seed. So we have these large-scale dynamics in gene expression. And at the same time, we also observed a very widespread demethylation of the genome, which would permit gene expression. And we observed large changes in the small RNA populations, which would be associated with changes in both expression and methylation. What we wanted to do was move forward from this observation that all of the genes change and explain how that is regulated. To do that, we used an approach called dynamic regulatory events mining. This approach depends upon the idea that genes with similar patterns of expression over time are likely regulated by the same transcription factors, the same proteins binding to their promoters and turning them on or off. And so this cartoon illustrates what this means. On the left-hand side, you see a cartoon of some gene expression data, some pretend gene expression data. Let me see if I can find my pointer here to make this a little easier. Okay, there we go. So you can see in this sketch here, we've got a group of blue transcripts, a big group that are co-expressed going up and coming down again. We've got a magenta group that all go up and stay up, and a red group that go down and stay down. The concept behind DREM is saying, okay, transcripts that exhibit similar patterns of expression are regulated by similar transcription factors. So our magenta group might be regulated by a transcription factor A. Our blue group might be regulated by a transcription factor B. And our red group, perhaps they're regulated by transcription factors C and D. And we want to find those transcription factors because this will tell us how these genes are regulated. So when building a DREM model, basically you look for transcription factors that are associated with one expression path or the other at split points in the co-regulation. And you do this by using both your time series gene expression data, which I've shown you, and then large-scale transcription factor gene interaction data. This would be something like ChIP-seq. Now, we need an awful lot of transcription factors worth of data to make this model work, and so we actually used a resource called DAP-seq. This was developed by Ronan O'Malley and Kara Wang in Joe Ecker's lab, and so these guys developed a really innovative method for high throughput in vitro ChIP-seq effectively. So they can take transcription factors, they can probe them against genomic DNA, and they can find all of the binding sites across the genome for that transcription factor, meaning all of the genes it binds to. And that some of that data is shown in an example on the left-hand side of the screen in this genome browser shot here. So with this, they were able to map the target genes of 529 Arabidopsis transcription factors. That's about a bit more than a quarter of the transcription factors encoded by Arabidopsis. This is then sufficient for us to build our model. So we can take the target genes of all these transcription factors, we can relate them to the changes in gene expression over time during germination, and we can say, okay, these transcription factors are associated with these patterns of change. And that is what we did for our germination study. This is how our model looks. So what you're seeing on here is germination progressing from left to right, 0 hours of germination through stratification and then into the light and warm temperatures. Each, the bars, the branches, so to speak, represents the average expression value of a group of transcripts, a co-regulated group of transcripts. And you can see that we reach these branch points where patterns diverge. So this particular set of transcripts carries on up. These guys over here don't go up as much. And at each branch point, the boxes label on transcription factors that are predicted to regulate that branch event. As an initial common sense check, when we did this, we could find pretty much every known germination regulatory transcription factor. So that's great. But what we get from this is actually an awful lot more transcription factors associated into control of gene expression. It illustrates the complexity of the control of gene expression during germination. And we find that the transcription factors involved are changing over time. So we have different ones in the early phase to the late phase. And so now we have a structured model about which transcription factors act at what points in germination and control which genes. So we built the model, but the acid test of any model is, can you break it? And are the outcomes in some way related to the phenomenon you're looking at? Are they in some way predictable? And that is what we did next. So we tested this model by mutating some of the transcription factors in the model. We selected 8 transcription factors and we tested them in 2 ways. We got tDNA mutants for all of our transcription factors. And in our first assay, we compared the rate of germination of the transcription factor mutants to a wild type. And in the second assay, we compared the transcriptomes, the gene expression patterns during germination in our mutants to wild type. So what you can see on the left-hand side of germination assays, for 7 of the 8 transcription factors that we tested, the rate of germination was delayed. 7 out of 8 were slower to germinate than a wild type. So mutation of the transcription factors affects those. And then when we look at gene expression, in all 8 of our transcription factor mutants, their patterns of gene expression are different during germination. So disruption of the transcription factors affects gene expression at the relevant stage. And so with this, we've now got a quite workable model and a host of new, useful, interesting candidates to regulate this process. However, as I said at the start of the talk, there is a limitation that is apparent in this. This was all done using bulk seeds, whole seeds, as we went through. I would imagine that most people are aware that a seed is actually a fairly complex structure, which is what I show you on here. So this is a section through an Arabidopsis seed, and you can see all of the different tissues within the seed. So you can see the cotyledons up here, you can see the hypocotyl going down into the radicle. You can see within those we have different tissues. So we've got epidermis, we've got provasculature, which will develop into the vasculature itself, a whole host of different seeds, different cell types. These tissues, these cell types, are defined by different patterns of gene expression, which are necessary for them to have their distinct functions. And so by looking at bulk seeds, although we've gotten a useful model for the system, we've lost a massive degree of resolution in it. When we did the study, it wasn't possible to analyze in this level of detail, but methods now become available to do so. I would point out, though, that the complexity is not just in the mixture of cells, it's in what those cells are doing over time. Here I'm showing you some work from George Bassel's lab. And what this is illustrating is that different cells are doing different things at different points in germination. Here we're looking just at growth in the hypocotyl and radical cells of a germinating Arabidopsis seed. And again, time moves down on this axis over germination. The figure shows you volume increase of the cells, radial increase and longitudinal increase. So assays of how cells are expanding at this time. Red is high, blue is low. So you can see across all of these tissues, different cells are growing at different rates depending on what the time is of germination. And so on top of the complexity of individual cell functions, we've got the complexity of cells even within the same tissue being at slightly different points, slightly different times during germination, getting started at different rates. In order to tackle this, we have been developing a single cell approach to analyze it. So single cell transcriptomics to analyze gene expression in each of the individual cells of an embryo as it progresses through germination. Our study looks like this. We have taken seeds at 3 time points, 12, 24, 48 hours of germination. And we're going to simplify our challenge by looking at just the embryos. So we're going to very carefully pop the embryos out of the seed coats. And the reason we need to do this is that in order to do single cell transcriptomics, we need to dissociate all the cells and pass them through a microfluidic device. And we can't dissociate the cells of a seed coat. And we actually also lose the small amount of Arabidopsis endosperm in this process. So we've got a purified enriched fraction of just embryos. And then we're going to dissociate the cells by protoplast isolation. We're going to degrade away the cell walls with enzymes and separate out all of our cells. And we end up with samples of protoplasts from our 3 time points, 12, 24, 48 hours. That's what we're going to use to do our single cell experiment. But before we do our single cell experiment, there's an important control that we have to do here. We've made quite a dramatic intervention when we prepare these samples. We degraded the embryo. We separated out all the individual cells. So we harvested bulk samples prior to protoplast isolation of just embryos, whole embryos, and then bulk samples of the protoplasts that we isolated. And what we want to do is compare gene expression between the whole embryos and the protoplasts, just to get a handle on how much is gene expression affected by this. And the answer is that protoplast isolation does have quite strong effects on gene expression, and doing this control is important if you want to do a single cell study. So when we compare our protoplasts with our embryos, our non-protoplasted samples, we find around about 2,000 genes are responding to that process. Helpfully, thankfully, it actually seems like the same genes, or it is that the same genes respond at each of our 3 time points, 12, 24, and 48 hours. There's a protoplast response gene set during germination. And so we can gray list, we can remove those from our analysis and say these genes are affected by our treatment, let's not analyze those. But I would like to point out that this is a control that people should certainly consider doing. So in the literature, the majority of existing Arabidopsis single cell studies actually lean upon a list of protoplast responsive genes from a study from Ken Birnbaum's lab a few years back, which I believe was on wound regeneration. They take the list of genes from there and they remove those from their particular analysis. But these new studies are not all done in exactly the same conditions, in exactly the same way, and the same developmental stage as the Birnbaum lab study. And so, for example, when we compare the genes upregulated during protoplast isolation in our study against the protoplast responsive genes from this list from the Birnbaum lab, they are not the same. We have about 1,400 upregulated genes. Only about 100 of those are shared with the ones from the Birnbaum list. So I caution those of you doing this kind of work to really do your own control and apply it to your data, because the response to protoplast isolation may vary depending upon the developmental stage you look at. Okay, so we've now done our control. We can move on with our single cell analysis, and we're going to use a 10x genomics approach. We're going to flow our protoplast through a microfluidic device. And what we've got are germinating seeds at 12, 24, and 48 hours. We've done this in biological duplicate, and this yields about 13,000 sequence cells, and we have the complete transcriptome for every single one of those cells. When we analyze them, and I'm skipping over, I'm going to briefly summarize a very long process here. What we do is we integrate the data from the different replicates. We look for consistency. We integrate the different time points as well. We then identify clusters of similar cells by virtue of the fact that they have similar transcriptomes. So cells with similar transcriptomes are likely the same cell type. And then we need to annotate cell identities. And this last point is actually the trickiest part, the hardest and most time-consuming bit. So when our data comes out and when we've done cluster identification, it looks like this. This plot, every point on it is one of our cells, one of our 13,000 cells, and the plot it's a UMAP plot. For those unfamiliar, it's analogous to a PCA plot. It isn't a PCA plot, but it's analogous to that. So every cell is separated by its transcriptional identity. Cells near to one another have similar transcriptional identities, similar transcriptomes, and cells further from one another have different transcriptomes. Computationally, we identify clusters that are coherent sets of gene expression, and we find about 15 clusters. This is actually not very useful. We have got a whole bunch of numbers and colors that are indicated on the graph, but we don't know what cell types they are. And if we want to interpret the biology, we need to know what the cell types are. There's a good reason that the vast majority of current Arabidopsis single cell papers are on the Arabidopsis root. And that is because Phil Benfey's lab spent the best part of a decade, I think, carefully pulling out each individual cell profile of the roots and analyzing the transcriptomes of them one by one. This provides a ground truth that people can use to compare their single cell data and say, okay, I know that this is a columella cell, and so I can find it in my single cell data. None of the rest of us have that benefit. We have to figure out what our cell types are in a different way. And so a couple of postdocs in the lab, Lewsey and Quentin, painstakingly went through hand curation of the literature to look for marker genes that correspond to the clusters we've identified. So one by one through papers saying, okay, this gene is characteristic of this cell type. So now we compare it back to our clusters. We find, okay, this is a cluster that highly expresses this marker gene. This graph, or this plot, illustrates what we've got after we've done that. So we were actually able to take 13 of our 15 clusters and annotate them with cell identities. Broadly, they're across all of the major tissues, so cotyledons, hypocotyls, radicals, provasculature, and then they're annotated actually down to particular cell types. So we can see mesophyll cells, cortex cells, epidermal cells, and so forth. So 13 of our 15 cell types have now got identities. Interestingly, we have two that you see labeled at the top over here, unknown cell types. We had two that they don't look like anything known. And these are perhaps the most interesting thing because they don't bear markers of any known cell type in a germinating seed, but they do bear characteristics that are very strongly associated with dry seeds prior to germination. And so they look like they're a very particular cell state that I'll return to very briefly later on. So we've annotated identities from the literature, and obviously we need to go back and we need to check this. We need to confirm our cell annotations. We did this using RNA in situ. And so what we did was take clusters that we'd annotated. We then found independent marker transcripts in those. One, transcripts that were definitive of the cluster, that represented the cluster, were tightly expressed there, but that had not been used to assign the identity of that cluster. When we do that, we get quite nice validation. So I'm showing you examples here of cluster 9 and cluster 13. So cluster 9, if I just move my little box, cluster 9 is annotated into the provasculature and the xylem cells particularly. When we do our RNA in situ to check the pattern of marker gene expression, you can see we get this lovely tight signal right here in the provasculature in what's going to become the xylem cells and here. So that's an Arabidopsis seed through two different planes. The second one is cluster 13. This was annotated as mesophyll cells of the cotyledon. And again, you see this lovely tight signal in the cotyledon in the mesophyll cells here and here. So this validates our assignment of cluster identities. It also illustrates an interesting point in the data, though. It illustrates the potential temporal nature of cluster identity. So you can see how for both of these marker genes, we detect the clusters at 48 hours of germination. But when we look at 24 hours of germination, there's really not a signal to speak of. And so it's as if the clusters are somehow changing. If we go back to our PCA type plot with all of our cells that we've isolated spread out based upon their transcriptional signatures, and we overlay the time at which that cell was isolated. So was it a cell from a 12-hour embryo, a 24-hour embryo, or a 48-hour embryo? What we can see is there's actually quite clear spatial separation indicating the cells are different between 12, 24, and 48 hours. So we've got 12 in the middle here, And then we've got 24 over here, and then moving out to 48 hours in a separate space. Now, you might interpret that as saying we've got different cells. The 12-hour cells are not the same as the ones present at 24 hours and at 48 hours. This, though, is not possible. As I said at the start of the talk, there's no cell division and there's no cell death during germination in the strict sense. Growth is all by cell expansion. So the same cells are present at 12, 24, and 48 hours. What this is suggesting is that actually the transcriptomes of the cells are changing. The transcriptome identity of the cell is changing and causing them to move across the plot across time points. This is even better illustrated by looking at the clusters that we detect over time in our experiment. So if we look at which clusters we detect at each point of germination, they change. This graph, this plot, is showing you the clusters we detect at 12, 24, and 48 hours, and it's telling you the proportion of cells within a cluster at any given time point. Just looking at it very crudely, you can see the colors change, which is indicating that the clusters we're detecting are changing. You can see, again, interestingly, our unknown cells that we aren't quite certain what they are, they constitute a large proportion of the cells that we detect at an early time point, and then they kind of go away. But if I focus your attention particularly on the hypocotyle cortex cells, this illustrates the point quite well. So we have 3 clusters that are hypocotyle cortex cells. We've got clusters 1, 5, and 7 in different shades of blue. Cluster 5 is the dominant cluster at 12 hours, but then clusters 1 and cluster 7 go on to be the dominant hypocotyle cortex cells at 24 hours. And then later on at 48 hours, we're really only detecting cluster 1. So it appears that our hypocotyle cortex cells are transitioning through clusters over time. And we can then zoom in and analyze and ask, okay, how are their transcriptomes changing that the clusters of hypocotyle cortex cells are different over time? So if we want to zoom in and understand how the transcriptomes in these clusters change, what we're going to do is repeat our PCA analysis to get a more fine-grained view resolution. We're going to get rid of all the cells that are not hypocotyle cells because they're very, very big differences, and they might cloud, they might hide differences we wish to see. And we're going to repeat the clustering and just have the hypocotyle cells on there. And so here we have our hypocotyle cortex cells. What we're looking to see is, okay, at this fine resolution, are clusters 5, 7, and 1 still similar? How are they similar? We find that the cells of cluster 5 largely form a group here that is kind of contiguous with the cluster 7 cells, that's kind of contiguous with the cluster 1 cells. It's as if our hypocotyle cortex cells form a kind of a spectrum or a trajectory that spans across clusters 5, 7, and 1. And each point is a cell, and each cell is positioned due to the identity of its transcriptome. So neighboring cells are very similar. Further apart cells are less similar. If we take this same analysis and we instead overlay time, you can see the time at which we isolated those cells, and you can see that this time forms a similar kind of spectrum or trajectory that goes across clusters 5, 7, and 1. So you start with the 12-hour cells at the top, and those seem to merge through the 24-hour cells that bridge down to our 48-hour cells predominantly at the bottom here, as if they're going along a trajectory. Using this, we can actually attempt to reconstruct that trajectory. So we can say, okay, we've got a 12-hour state passing through a 24-hour transcriptome state to a 48-hour transcriptome state. How does it change? And we can do this using an analysis called pseudotime. Pseudotime essentially attempts to sort all of the cells onto a continuum and look for a path through their transcriptomes, through the changes to go from one state to another. So it's trying to go from 12 through 24 through 48 hours through changes in the transcriptome. And to do this, it takes advantage of the fact that the cells are actually a little bit asynchronous. They're not all going to wake up in germination at the same time, and so they're not all at the same point on this transcriptome trajectory. It sorts them into an organized continuum. And the reason this is useful, because we're diving quite deep in the data, the reason this is useful is that it sorts the cells into a defined order across this trajectory, which we can then use to say, okay, what are the characteristics of the hypocortical cortex cells early in germination versus the middle of germination versus late in germination. So we find our trajectory that it follows the same trend. 12-hour cells are up here. It passes down through the 24-hour cells and ends up in the 48-hour cells over here. And it also illustrates a little bit more complexity with a branch that's coming in over here from some cell state that must exist there. But we have this trajectory, we have our cells ordered, and we can now say, okay, what are the characteristics of early, mid, and late cells? What's happening? How are they different? To ask that question, we essentially do a form of co-expression analysis. We look for genes that are particularly expressed in groups, cohorts, modules, early in germination versus mid versus late in germination. And we find a sequence of several groups of co-expressed genes with particular functions. So we have on here, this is a heat map illustrating the trend in expression of these cohorts of gene expression. We have an early group of co-expressed genes, and they seem to, or they do, when we analyze the function of those genes, express things that are related to regulation of transcription, to turning on gene expression, and to reorganizing chromatin. So those early events you would need when waking up the nucleus. In the middle, we see genes that are associated with translation, protein folding, and also the integration of some signaling pathways, things like abscisic acid. And then late in our pseudotime, which is late in germination, we see events related to photosynthesis and the chloroplast and reorganization of the cell walls. And so those, particularly the chloroplasts, are things that happen late in germination. And we can actually make use of this organization of the data to ask, in a similar fashion to our DREM models at the start, which transcription factors regulate these processes. And so we find that there's actually a very short list, just 10 transcription factors that are associated with regulation of these particular genes at these particular times in just the hypocortical cortex cells. So we find some ABA transcription factors in the middle phase. We find some cell wall transcription factors in the late phase. And so that is an incredibly deep dive on one very specific cell type that's in the middle of the embryo during germination. And you might all reasonably ask, what's interesting about that? That's really getting into the nitty-gritty. And quite honestly, this individual cell is not super interesting. That's kind of not the point here. The bigger point, the bigger picture that comes from this is that these dynamic processes will be occurring in every individual cell type and in every cell type differently across the entire embryo going through germination. And in fact, you can apply that same thinking to any organ you happen to be looking at, different cell types within it, in the plant that's responding to a stimulus. All the cells are doing something different at a different time. And so our job going forward is to make use of these data to understand how that regulation varies between the different cell types over time, what's different about one cell type versus the next, and particularly to understand this unknown cell type, the two clusters we couldn't annotate that actually appear to be a quite distinct, uncharacterized cell state that is occurring at the start of germination. So with that, I will wrap up. What I've told you today is that substantial remodeling of gene expression occurs during germination, and this is controlled by a suite of transcription factors that change over time through this process. But then I've told you that that kind of approach is somewhat limited, and in fact, seeds are very complex structures composed of many cell types. And so we've used single cell methods, and from this, we find that the transcriptomes of individual cell types change during germination, reflecting changes in function across the period of germination. So all that remains for me to do is actually thank the people who did the work. So I must point out the first study was Quentin, and Rena particularly led this. And then the second work, the work in progress I've described to you there, is Limchi and Quentin together with Yue, a very talented grad student over at the Walter and Eliza Hall Institute. We've had great collaborators in Jim Whelan's lab, in Marnie Blewett and Matt Ritchie at Wehi, and in Ryan Lister's lab over at UWA. And getting a photo of everyone over the last 2 years in the same place at the same time has been incredibly difficult, and more so now that I am not even in the country. But we did have this very nice socially distanced cake with everyone's picture on it. You can see me at the top, and I really was delighted they illustrated my hairline accurately. But I'm sure you'll all recognize Limchi from this character here when you meet her at a conference or something. And thank you all for your attention.
Thanks very much, Matt. That was a fascinating talk. For those in the audience, if anyone would like to raise their hand to ask Matt a question, or please put it in the chat box on the Q&A and I will read it out. But to get the ball rolling, so one of the cool results from the DAPSeq paper you mentioned, Matt, was the finding that the majority of transcription factors in Arabidopsis had methylation-sensitive binding. From the first half of your talk, looking at your predicted transcription factors regulating genes during germination, did you find many of them binding sites at which there was differential methylation? And was there any evidence of an interplay between those two during germination?
Yeah, I'm just thinking back now. So I think we definitely did. I think it was maybe like 20% or so of binding sites exhibited some differential methylation. There was one particular family of transcription factors, which I apologize, I forget the name of, but there was one particular family of transcription factors that seemed to be associated with these changes in methylation. Honestly, we did not find a statistically significant relationship between the gene expression and the methylation, but the changes were massive across the entire genome. Is basically passively demethylating. It's not an active process. It seems to be as endoreduplication occurs, the methylation is lost and not put back properly. And so there's just this widespread demethylation of the genome, and about 20% of promoters are losing methylation. So we think there's a relationship there. I think this is actually another thing you need to get to the single cell level at, because I don't think demethylation is happening at the same rate in all the cells. But that's a whole new can of worms to get into.
That was another one of the questions I was going to consider asking. So we have a couple of questions in the Q&A. So an anonymous attendee, how much does the pseudotime cell-specific transcriptome improve upon raw time point sample transcriptomes?
So the order does actually help us. So what's going on is that the cells are not all passing through germination at the same rate. And also bear in mind, we've got replicates we're trying to integrate in order to understand or get statistical power over the data. And so you will find cells from different clusters popping up at more than one time point, but they're changing in number. So the pseudotime ordering does actually make it easier for us to understand what are the early events versus the late events. It allows us, essentially to sort those clusters into an order rather than just say, oh, cluster 5 is the early time point, 1 is the middle time point, and 7 the late time point, something like that.
All right, and a question from Caitlin. Awesome talk, Matthew. Very powerful approach you developed. Wondering if you've published a report on how protoplasting changes expression, i.e. what exactly changes. This would be a great resource, and I definitely agree.
Yeah, so that's all going in. I hope to get this finished by the end of the year. We still, as I say, we have work to do on it, but I hope to get this thing out there by the end of the year. Actually, I think that, so one person who has done this properly is Ryan Lister, who we learned the single cell approaches from. And if you have a look in his preprint, which is already up on bioRxiv, I suspect they've included their protoclast list, which they did for their specific root experiment. So you could start there. But yeah, we will put out our list of the seed response genes when we publish this for sure.
Cool. Another question I had was the large number of changes in RNA abundance you observed during germination. Do you think that's mostly a transcriptional response, or do you know if some of that might reflect a post-transcriptional regulation of, say, seed-stored mRNAs that occur during germination?
Yeah, so I think this would be really interesting. We just don't have the data to fully get at it. So you can see for sure that transcripts are stored, they're there at the start, and they must be being degraded away somehow. So you've got some RNA processing and turnover going on there. And if you knew what was binding to and degrading those, you could actually include that in your DREM model. So if you've got RNA binding proteins that are particularly responsible for removing certain RNAs, the DREM approach isn't dependent upon binding to a gene. It's just an association between the level of a transcript and a transcription factor or any other entity. So let's say we could take all of RNA binding proteins in the cell and they're involved in degradation of RNA, and we could map those to individual transcripts. We could roll those into the model as well, and we could predict which RNA binding proteins might be removing transcripts and degrading them away. But that kind of data set doesn't exist. There's not like an RNA degradation DAP-seq that we can use to do it. If someone was to build that resource, it could totally be done.
Cool. A question from Kai. Hi, Matt. Great talk. What are your thoughts about adding additional transcriptional inhibitors to the protoplasting medium to eliminate or reduce protoplasting effects on the transcriptome?
Oh, that's an interesting suggestion. Yeah, I think you could do that. We did not think of that at the time. That is actually a very good suggestion. I think you could do that. We're doing this. We can do this pretty quick. We make our protoplasts and get them into the single cell device within about an hour, and we're doing it at a very low temperature to minimize changes as much as possible. But you can see there are changes. And I think inhibiting transcription would be helpful. You'd still have the issue around RNA degradation, I guess, but it would reduce the effect, I think. Yeah, good suggestion. Thanks.
And I had another question around the analysis of the single cell transcriptomes and some of those UMAP plots. To what extent are noisy, low abundance transcripts influencing that analysis? And do you have issues around like different cells kind of having presence and absence of different transcripts in different cells that may complicate the ability to actually look at relationships between different samples? And how did you get around that?
Yeah, so single cell data is plagued by being sparse. You get a lot of dropout of signal. And so in constructing those plots, we're doing a lot of screening out of low abundance and low quality measurements. And we're focusing on the things that we can more reliably make estimates of. Bear in mind though, like they're essentially very heavily replicated because we've got 13,000 cells and you've got many cells of any individual cell type. And so this is helpful to you when trying to construct it, if you see what I mean.
Yep, gotcha. And another question from Martin, very interesting and cool approach. Have you considered using the DREM approach on translational processes using polysome-associated mRNA and RNA binding protein databases?
No, I have not considered that, but a bit like we were discussing just a moment ago with the degradation, again, I think the DREM model is essentially agnostic. It's just a measure of interactions between proteins and genes. It doesn't matter whether it's promoters, it's poly-A binding or whatever. So I see no reason it could not be done.
Cool. Well, in the interest of time, we might leave it there. Thanks so much for joining us so early, Matt, and sharing a wonderful talk. If I could ask all the attendees to join me in thanking Matt by using the raise hand function in unison. Thank you, Matt.
Thanks very much, guys. It's been a pleasure.
And hope everyone has a pleasant evening. Thanks all. Let me just find the stop.